everyone. My name is Emely Henriquez Pilier, and I worked in Dr. Alice Stanford's lab this summer. And our research was on the genetics of the invasive seagrass Halophila stipulacea. Seagrass play an important role in our economy. They stabilize the sea floor, provide food and habitat for sea organisms and creatures. They can convert CO2, stop runoff from the land, and so much more. And many things can threaten the local seagrass, whether it be from on land or in the sea. One such thing is the invasion of a new seagrass. The seagrass I'm talking about is the invasive seagrass Halophila stipulacea. It originated in the Indian Ocean, and when the Suez Canal opened in 1869, it migrated into the Mediterranean, where it was then able to cross over the Atlantic Ocean and enter into the Caribbean. It was found in Grenada in 2002 and then propagated throughout the region and was found in St. John in 2012 and now in the other islands such as St. Thomas. Here is a picture of the invasive seagrass. It's usually seen with two mid-sized leaves, rhizomes, roots, etc. And the thing about this seagrass is it's a fighter. It's able to survive different light intensities, different water temperatures, salinities, and even in unfavorable conditions, this seagrass is shown to thrive. And what this seagrass does is it sees any small, open patch, and then it goes there. Then it propagates. It takes all the nutrients away from the local seagrass and the space from it, and then the local seagrass ends up dying, and this seagrass just ends up taking over. And some might say it's just a seagrass, and the more the better, but our local organisms don't seem to like it. They like their native seagrass, so they're not eating this invasive seagrass. And what this can do is the native seagrass is going to decrease because they're continuing to eat it while the invasive seagrass continues to increase. And this will cause a problem in the future because if they don't take to this new invasive seagrass, they won't have any more seagrass to eat because their native seagrass is being depleted. So that's definitely going to be a problem. And many people are looking at different things such as how the environment is affecting this seagrass, but what we looked at was the genetic importance, because we think genetic structure of seagrass can be key to their functioning. And this project was started in 2016. So we are testing for genetic structure and clonal variation because it can tell us if this seagrass is still being introduced to our islands, or is it still coming in. It can also tell us, is this seagrass, is it just the same seagrass that's already here, just spreading out throughout the different islands? So our questions were, what can genotypes tell us about the invasion of Halophila stipulacea? And since this project was started in 2016, we wanted to know, were there any differences in extraction quality throughout the various years? Because throughout the different years, there could be differences. And our hypothesis was, once Halophila stipulacea invasion occurs, expansion is not related to genotype, and that there won't be any differences in extraction quality between the different years. So my main role was to confirm / replicate the data that was found in the previous years. So our methods were that the samples were collected in 8 different sites from St. Thomas, St. John, and Water Island. The DNA extraction process was started, which is breaking down the DNA and all that. And then PCR was done, which stands for polymerase chain reaction. And if you've taken a genetics class, you know this is a way to make millions and billions of copies of a DNA sample. And then we did DNA sequencing. So my progress so far is that I did all these samples that were from old leaves that were dried and stored, and primarily 2016, 2017, etc. And These samples will then be sent to the states, a lab in the states, to be analyzed, and then that data will come back to us where we will analyze it further. And so I haven't gotten the data back yet, but this is pretty much a graph of what we would want with the data. So we use GenClone, which is a computer program to analyze genotypic data, test for clonality, and describe spatial clonal organization, so how far away these clones and genotypes were from each other. So on this picture, you can see the 8 different sites collected: Brewers Bay, Megan's Bay, Lindbergh Bay, etc. And over here are on the left are the different genotypes found. There were 16 found in that year. So they were labeled from 1 to 16. And it can also tell you that you can see that genotype number 1 was the most abundant. It can also tell you how far away they were from each other. So like genotype 10 and 11 were very close to each other. So that could be something to look at, like what was occurring there. While genotype 5 and 9 were very far from each other. So might probably not be too related to each other, but Lindbergh Bay seemed to have a lot going on. So maybe there's something going on the west side of this island. So what this can tell us is that it probably is multiple invasions, or maybe somatic mutations. So it's multiplying, it's mutating very quickly. So we also checked the extraction quantity for various years, and I chose 2016, 2017 because those had the most data, and summer 2020 is the year that I worked on this research. So I want to check, was my extraction good enough? So it was found that there was an ANOVA was done, and it was found that 2017 was significantly different from 2016 and summer 2020. As you can see, it's all the way up here, like at 39 nanograms per microliter, while 2016 and 2020, there were no significant difference between the two. So 2016 was the year the research started. So the amount could be because new protocols were being created, different things were being tested, and trying to understand the whole research process. So that's why maybe this was low, while in 2017, things are pretty much already established, understood, and going well. So that's why this probably had a huge difference. And what's great is that my summer 2020 didn't really have much different from 2016, even though I was only testing old leaves that were stored. So 2016 we found a lot of data that year. So summer 2020, my data could show us a lot, and it can be used in analyzation. So I would like to acknowledge that I was in NIH Mark at UVI, and this is my email if anybody wants it. And I would also like to say that thank you.